Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that's your microbiology of butter. Okay, we all know that this butter is what? Butter is a dairy product. Butter is a dairy product, which means that it is made up of or manufactured by a milk. Okay, now this microorganisms are microorganisms are present in everywhere. Okay, and it cannot be seen by our naked eye. So it is obvious that this butter will also contain the microorganism. Okay, now here what should we are going to do? We are going to do the discuss about the microbiology of the butter. Means that the types of the microorganisms which are present into a butter, we are going to discuss about this, because the microbiology is what? Microbiology is simply study of the microorganism. Study of microorganism. But here what should we are going to do? We are going to discuss about the microorganism. We are going to discuss about the microorganism which are present in a butter. Okay. So either maybe they are beneficial microorganism. Okay. And either they are harmful. Okay, so these both type of microorganisms like beneficial and harmful both will be discussed in today's topic. That is your microbiology of the butter. Okay, so let's start today's session. First of all, we have to know that what is this butter. Okay, when we are going to start anything, we have to know about it. As early, I told you that butter is what? Butter is a dairy product. Okay, and simply it is manufactured by a churning the cream or milk. Okay, when we churn the milk or a cream, then this butter is formed. Okay, now this butter is what? Butter is a water and oil emulsion. Okay, and it contain around 80% of a fat. Okay, if a product does not contain minimum 80% of the fat, then it will not consider as a butter. Okay, so if we said that this product is a butter when they contain around 80% of the fat. Okay, so water level is around 15 to 17% and carbohydrate is around 0.5% and this protein is around also 0.5%. Okay, so this butter is also come under the category of high caloric food, because if we talk about 100 gram of this butter, then it will contain about 700 kilo calorie. Okay, and they will also rich in different type of the macronutrients, that is your minerals like calcium, phosphorus and potassium. Now see what is written in a slide. Butter is a dairy product made by churning fresh or fermented cream or milk. Okay, either what should we do? Either we churn the fresh cream or either we churn the fermented cream or milk. Okay, butter is what? Butter is a water in oil emulsion form. It consisting at least 80% fat, 15 to 17% water, 0.5% carbohydrate and protein. Butter is either as a high calorie food. 100 gram of butter provide almost 700 kilo calorie. It is extreme in minerals like calcium, phosphorus and potassium. Okay, now the next one is your, we all know that it will also contain like sodium, some small amount of fluoride, selenium, zinc and magnesium, and they are what? They are a metal ion. Okay, so the microbial quality of the butter is totally depend upon the microbial quality of the cream. Okay, so if we talk about the microbiology of the cream, the video related to this are already uploaded in our account. You can go and check it. That is your microbiology of cream. Microbiology of cream. Because the microbiology of the butter is totally depend upon the microbiology of that cream. Because this cream is what? Cream is a primary ingredients for the manufacturing of the butter, and this butter should be prepared in a hygienic environment, and it will produce a good quality of the product. Okay, so butter have also some intrinsic properties like its pH, its salt content, its moisture distribution. Okay, so these are what? These are their intrinsic property. Now see what is written in a slide. Good amount of metals like sodium, and small amount of fluoride, selenium, zinc, and magnesium. Microbial quality of butter is primarily depend on cream, as it is the primary ingredients used for the butter manufacturers, and the hygienic quality of the production. Intrinsic properties of butter, for example, pH, salt content, uniformity of moisture distribution. Now the next one is your definition of the butter. Okay, so here the introduction part of the butter are completed. Now what is the definition of the butter? We all know that this butter are what? Butter are defined as a fat concentrate which are obtained by a churning of the cream, and the when the cream are churned, then their fats are gathered and converted into a compact mass, and this is called what? This is called a butter. But if we talk about according to the FSSR, okay, now again here one question for you. FSSR stand for. Now write your answer into a comment box. Okay, what is this full form of this FSSR? Now, if we talk about according to the FSSR, then they define that the butters are simply what? Simply they are a fatty product, and it is derived from a milk. Okay, milk of cow, milk of buffalo, and its product are principally form a water in oil emulsion. Okay, so this product is either they contain salt, okay, and either they does not contain the salt or a starter culture or a harmless lactic acid. It totally depend upon either they will contain it or either they will not contain it, because there are some butter or also present in a market which are not contain the salt, which are not contain even the starter culture. Okay, starter culture means that with the help of this, we start the manufacturing of the butter. With the help of this, the manufacturing of butter start. The manufacturing of butter start. Now see what is written in a slide. Butter is commonly defined as a fat concentrate which is obtained by a churning cream, gathering the fat into a compact mass and then working it. As per FSSR, now, according to the FSSR, butter means that the fatty product derived exclusively from milk of cow or buffalo, or its product principally in the form of an emulsion of type of oil, water in oil. Okay, the product may be with or without added common salt, starter culture, or harmless lactic acid or flavor producing bacteria. Okay, now the next one is your as per codex in standard. Then, like according to the codex in standard, the butter are what? Butter is a fatty product which is formed due to the emulsion of water in oil. Okay, butter is what? Butter is a fatty product in the form of emulsion of type water in oil. And this definition is according to the Codex standard. See, there are different standard which give different definition. Like you see earlier that FSSS give a different standard, different definition, and here the codex are also give a different definition. Now, if we talk about the butter and their compositions, okay. So if we talk about the table butter, then see, this is the first thing is your product name. Then here the moisture content. Then here a milk fat. Then milk SNF. Then common salt. Okay, if we talk about the table butter, then it will contain uh, their moisture content should not be more than 16%. Okay, and milk fat should not be less than 80%. SNF should not more than 1.5%. Okay, and common salt should not more than 3%. Okay, now desi cooking butter, the butter which we cook in our home. Okay, then here are no data for a moisture content, but their milk content should not less than a 76%. And here there is no data for us SNF and no data for a common salt. It depends upon if you want to add a salt, then you will add it, because this desi cooking butter are manufactured in a home. Now the next is your microbial standard of butter. Okay, so here the different microorganisms and their quantity present in per gram of butter. Okay, if we talk about the total plate count, then it will contain around 10,000 total number of this plate count. Like, see, when we talk about 10,000, that means that CFU, 10,000 CFU, that is your colony forming unit, because it is count in this form CFU. Okay, so means that it contains 10,000 CFU per gram. What total plate count? Okay. Now the next one is your coliform. Then it will be at 10 CFU per gram. Okay. E. coli, it should be absent in 1 gram. Okay. Salmonella should be absent in 25 gram. Salmonella aureus should be present around 10 CFU per gram. Yeast and mold count around 20 CFU per gram. And spore count should be absent. And Listeria monocytogenes should be absent in 1 gram. Now the next is your we all know that butter is what less satisfactory for the growth of the microbes, because they have less lactose concentration and they have less moisture content. Okay, see, this lactose are what? Lactose are food, because they are what? They are a carbohydrate. Okay, and the amount of this lactose are also low in a butter. That's why it is um not the happen the growth of the microorganism, because we all know that this for the growth of the microorganism this factor are required. Okay, so food, if the amount of the food is less, then obviously food or nutrient, if the amount of the nutrient is low, then obviously the concentration of the bacteria will also low. Okay, now the next one is your moisture content. We all know that the moisture content of the butter is around less than 16%. Okay, and when the water concentration is low, then the chances of the microbial growth will also decreases. That's why these butter will should not help in the growth of the microorganism. Now see what is written in a slide. Less lactose. Many of the microorganism readily utilize lactose. Lactose, a disaccharide, is present in a lower quantities in butter, as most goes into a buttermilk, because when buttermilk are separated from the butter, then most of the lactose are present in a buttermilk. Less moisture. Moisture is an essential criteria for a microbial growth. It present lower quantities around 16% in butter, Water activities is around 0.947. Now the next one is your dispersion of water. Okay, fat is present in continuous phase and water is in discontinuous phase, present in the form of fine droplets. Okay, that's why the moisture content is low. Moisture content is low. See why this moisture content is low? Because we all know that fat and water are present because they form what? Water in oil emulsion. Water in oil emulsion. Okay. Now this fat are present in a continuous phase. Suppose that this is your butter. Now here in a continuous manner the fats are present. But the droplets of water are present. Due to this, the dispersion of water is low and the moisture content, and the microbial load will also low. Salt contains, we all know that salt is used as a preservative. Preservative. Okay, means that which kill the microorganism. Kill the microorganism. Now the salt are present in a salted butter and the salt are inhibit the growth of the microorganism due to the action of the preservative. Okay, now the next is your microstructure of the butter. We are going to discuss about the microstructure of the butter. There are several fat globules still largely undamaged. Okay, so we all know that the fat globules are present in a butter and they are undamaged. Now this fat globules in a butter poses a outer crystal shell. Okay, suppose that this is your fat butter. Now here a outer crystal shell are formed whose thickness are in between the 0.1 to 0.5 micrometer, which consists monomolecule triglyceride layer about 4 to 5 nanometer, which withstand the shearing forces during the churning. A large amount of droplets which are free from microorganisms are also found. Okay, so here are large number of the droplets which are totally free from the microorganisms are also present in a microstructure of the butter. Now, this is mainly due to the higher number of droplets than the number of microorganisms in a butter, because the number of the droplets are higher as compared to the microorganisms in butter. That's why the droplets are seen. As per water phase is separated by a relatively resistant fat phase in butter organism which cannot be grow. Now this is yeast and mold, however, are able to grow and penetrate through the fat phase of a butter. Okay, so here the chances of the yeast and mold growth are more because it will penetrate inside the butter through a fat phase. Secondary, there is a limited supply of nutrient in water droplet in butter, while in cream, microorganisms can grow in the continuous water phase, having a dissolved nutrient and migrate easily from one portion to the other. If we compare the microbial flora of butter and cream. Now we all know that in a butter the supply of the nutrients are very low in a water droplet, but in a cream the water are in a continuous phase. In butter water is in discontinuous phase. Okay, that's why the chances of the nutrients are less in a water droplets. Okay, but in butter the water sorry in cream the water are in the continuous phase and the nutrients are dissolved into that phase and they are migrate easily from one place to another place. Now this is the diagram of the water droplets. See, this is your water droplets. See, this is the microstructure of the butter which we are seen in a microscope. They are what? These are the water droplets because these are the discontinuous phase and the continuous phases are what are the fat globules. Now the next is your effect of various processing step during the manufacturing of a butter. Now the various processing step, how they affect the manufacturing of the butter, we are going to discuss about it. So for the manufacturing of the butter, first thing that we required is the milk. Without milk, the butter manufacturing is not possible. Okay, but here there are some butter are also present in a market which are plant based. Okay, they contain a plant-based product and they form a butter. Now the when we choose a milk, then the quality of the milk should be good quality, because it totally influences the quality of the butter. Because if we choose a bad quality of the milk, then the quality of the butter will also bad, due to the because this milk is what? Milk is the initial ingredient of the butter manufacturing. Okay, so a high quality of raw milk produce a high quality of the butter, and this milk which are used for a butter preparation, when after heating, it should be cool at 5 degrees Celsius. Okay, and it should be done after milking. Like suppose that milking are done, milking, then what should we do? We heat it, and then again we cool it at 5 degrees Celsius. Okay, and it should be need to be separate as soon as early possible. Now see what is written in the slide. Quality of the milk, quality of the butter is influenced by the initial quality of milk is used. High quality raw milk yield high quality butter. Milk to be used for butter preparation should be cooled to 5 degrees Celsius immediately after milking, and needed to be separated as early as possible. Now the next is your cream. Okay, now the cream which is used for the butter manufacturing should be fresh in microbiology, and they also have a high grade. Then the neutralization, commercial neutralizers should be used, like high standard and superior quality. If poor quality of water are taken, then their solvent also have a different constitute of bacteria, and ultimately it will impact the butter manufacturing. Now see what is written in a slide. If we talk about the cream, then always fresh in microbiological high grade cream has to be used for a butter manufacture. Neutralization, commercial neutralizers to be used must be high standard and superior quality. Poor quality of water should not be used as a solvent, which may constitute bacteria. Okay, when, because water is used for the neutralization, so we have to take care that we are not going to use a poor quality of solvent, because they will also have contain some constitute of the bacteria, and this bacteria will transfer to the butter and decreases the quality of the butter. Heat treatment. Now when we have a cream, then this cream should be pasteurized between a temperature range 85 to 95 degree Celsius. Okay, and the time around 50 to 30 seconds, means that 85 to 95 degree Celsius temperature, and the time should be 15 to 30 second. Okay, this is the time, and this is the process of the pasteurization, and this pasteurization is done for the killing of the pathogens. Higher temperature may lead nutty flavor due to the sulfihydryl compound. Okay, so we are here, you see that we are not going to give our higher temperature like more than 95 degrees Celsius. If we give it, then our nutty flavor are occur due to the sulfihydryl compound, and intensity of this nutty flavor may decreases with the time, and maybe the intensity of this nutty flavor will decreases when the time is passes. The thermal treatment has another advantage, that is to minimize the reactivation of the lipase native to milk, apart from the primary objective, which is to make a cream free from pathogen and spoilage organism. Okay, so the primary objective of this, what we have to make a cream free from the pathogen and spoilage microorganisms. Aging. Now this cream should be cooled between 10 to 11 degrees Celsius immediately after the heating. Like suppose that when we heat it 85 to 95 degrees Celsius temperature for 15 to 30 seconds, then immediately we have to cool down this temperature, and it should be reaches between 10 to 11 degrees Celsius, and the time should not more than 4 hour. Okay, within a 4 hour its temperature should be decreases to 10 to 11. Okay, this will allow a extensive fat crystal network. This process known as a aging. Okay, if the time is increases, then there is a chance of that fat crystal network will not form. Okay, that's why within 4 hours we have to decrease its temperature 10 to 11 degrees Celsius. This step allow to stable matrix of the fat crystal to develop, which is important for the physical properties of the final product. Okay, so that's why this step is very aging step is very important, because the physical properties of the final product, it's totally depend upon this. Now the next is your ripening. Okay, so ripening of a butter manufacture is carried out so that the flavor of the butter should improve, okay, of the product by utilizing a specific organism that utilizes lactose and citrate, yielding the flavoring compound diacetyl. It is a if the butter is to be ripe, high heat treatment sufficient to remove a thermoduric, enterococci, lactobacilli, and micrococci should be carried out. Now the next is your the heat treatment is additionally denatured the milk protein that will release a sulfuryl compound. Okay, if we give a heat treatment additionally, then the protein present in a milk will denature, and when the protein are denatured, it will release a sulfuryl compound. Okay, and this provide a better medium for a starter growth. Immediately after heating cream, it should be cooled between a 19 to 21 degree Celsius. Now ripening is carried out generally using a four method. Either we add a starter, or either we use a concentrate starter, or either we direct addition of starter culture during a butter churning method. Okay, and addition of starter distillate. Okay, and this will direct addition of starter culture during a butter churning method contain a NISO method that we discuss in a letter. NISO method. Okay, now we have to discuss it in a letter. Now the next one is your addition of the starter. Okay, so first one is the addition of the starter. Now the ripe cream for the manufacturing of the butter has been traditionally done by inoculating the pasteurized cream with a pure strain of LAB. Okay, lacto. Okay, that is your lactic acid bacteria. Okay, so the starter culture are either a mixture of acid producer, that is your Lactococcus lactis subspecies lactis, and Lactococcus lactis subspecies ceromoris. Okay, so this is the starter culture which we inoculate in a pasteurized cream, and it is a pure or either a mixed strain of lactic acid bacteria. That is your lactic acid bacteria. Okay, so when the starter are consist either the mixture of acid producer, that is your Lactococcus lactis subspecies lactis, and Lactococcus lactis subspecies ceromoris. Okay, now the next is your diacetyl producing species. Okay, so next species is what? Diacetyl producing species, that is your Lactococcus lactis biovir, diacetyl lactis, Leuconostoc mesenteroids subspecies ceromoris. Okay, now the final acidity of the cream should be at the range of pH 5.3 for a mild ripe butter, and 4.5 for a strongly flavored butter. Okay, if we want to produce a mild ripe butter, then their pH should be at the range of 5.3. Okay, final acidity of cream. I'm talking about the final acidity of the cream. But if we want to prepare a strong flavor butter, then the pH should be at the range of 4.5. Okay, see, it is more acidic. More acidic. Now the next is your starter concentrate. The concentrate form of butter culture known as a starter concentrate. Okay, when we use a concentrate form of butter culture, then it is called what? It is called a starter concentrate. Now increasingly used for ripening of cream due to a several advantages. Now these advantages are what? Butter prepared with a starter concentrate has been found to be equal or superior to the analog product made with a ordinary starter. Okay, when we prepare a butter with a starter concentrate, then it is either it is superior to the analog product, or with an ordinary starter. Okay, it also saved the period required for the propagation of the starter culture. Okay, so then for the propagation of this starter culture, the time will decreases. More uniform flavor is obtained because concentrate is produced under a more controlled condition. Okay, so the flavor which are produced inside the butter will distributed uniformly. Disadvantage: concentrate has to be stored at a lower, less than 10 degree Celsius or sub-zero temperature. Any fluctuation in temperature of storage may lead to the loss of the activity and viability of the cell. Okay, if we are not uh, if we are not store this butter which are produced by the starter concentrate, then maybe there is a loss of the activity and viability of the cell occur. Now the next is your direct addition of starter culture during a churning. Buttermilk, which is obtained as a byproduct during a butter manufacturing, has a significant market value. Okay, when we are producing the butter manufacturing, now they will contain two compound. One is your butter and one is your buttermilk. Now this buttermilk will also have a significant market value. Okay, sweet buttermilk has higher value than a sour buttermilk. Okay, so again the buttermilk which are produces is either a sweet buttermilk or either it is a sour buttermilk. Okay, so the value is high of the sweet buttermilk as compared to the sour buttermilk. Buttermilk obtained after the ripening of cream will be sour, and hence its market value decreases, causing a loss to the industry. To overcome this problem, method has been developed. So now the next is your one such process is a NISO process. So which problem we have to overcome? That the sour buttermilk have low market value. So for the overcome of this problem, the NISO process are developed. This NISO is what? This is the Netherlands Institute voor Zwiebelon Onderzoek. Okay, which is increasingly being used to produce a ripe cream butter. Okay, so this process was invented at the Netherlands Institute of Dairy Research. That's why it is called a NISO process. Okay, so the next is your, in this process, after churning a step, lactic starter permeate around 0.6%, which contain around 11 to 12% of lactic acid and whose pH is around 3%. And the aromatic starter culture of 2%, which are rich producer of diacetyl, are worked directly into the butter, and the butter pH should be reaches around 5.3 due to the presence of this lactic starter permeate. Okay, now aromatic starter added, which produces around 1.5 to 2.5 milligram per kilogram of diacetyl in the final product. Now this addition of this diacetyl will impart the flavor and inhabits the presence of this gram negative bacteria and fungi. Okay, when diacetyl are added, then it will impart the flavor and inhibit the growth of the gram negative bacteria and fungi. Now the next is your preparation of the starter permeate. Fermentation of partially delactulosed whey or other suitable media which contain milk components by a lactic acid bacteria continue for 2 days at 37°C. Okay, now this medium is ultra filtered, which is used to remove the protein and bacteria and to further concentrate the medium. Okay, so the next is your during the ultrafiltration. When ultrafiltration are done, then the macromolecules are removed. Macromolecules means that the proteins and carbohydrates, because fats are present, okay, are removed and concentrate in the retentate, whereas a low molecule weight solute pass through a permeated stream. Then the pH of the butter with the permeate from this process is more easily adjusted in the desired range, and it will reaches between a 4.8 to 5.3. Now permeate should be stored at low temperature, that is around 4°C, and it should be stored more than 4 months under a proper condition. Okay, if the temperature will not changes, then it should be stored more than 4 months without any spoilage. Okay, that's why the storage temperature are very important. Now the next is your starter distillates. Starter distillates are made from a fermentation of skim milk or non-fat dry milk. Okay, this starter distillate are either made up of the fermentation of the skim milk, or either it is prepared by a non-fat dairy milk, non-fat non-fat dry milk, not dairy milk, it is dry milk, by our several bacteria. Okay, see this is the starter distill are also considered as a grass, that is your generally recognized as safe by our FDA, Food and Drug Administration. And the culture which contain in a starter distillates are what? They are Lactococcus lactis species, lactis, Lactococcus lactis species Saccharomyces, Lactococcus lactis species Lactis bivalve diacetyl lactase, Leuconostoc citrovorum, and Leuconostoc dextrinium, dextranicum. Okay, now the next is your the ingredient contain more than 98% of water, and reminder is a mixture of butter like flavoring organic compound. Okay, besides of this diacetyl, starter distillate contain minor amount of acetaldehyde, ethyl format, ethyl acetate, acetone, ethyl alcohol, 2-butanone acetic acid, and acetone. Especially this starter distill are popularly used in the manufacturing of the dairy products. Now the next is your production of the bacterial distillates. Okay, so natural butter aroma distillate which can be used for the preparation by growing the mixed culture which consists of either Lactococcus lactis species lactis, Lactococcus lactis species Saccharomyces, and Lactococcus lactis species Lactis bivalve diacetyl lactis in the culture medium. okay, which contain in a skim milk and weigh as a substrate. Now, water vapor distillation is carried out effectively, which are in the presence of the oxygen, and the resultant distillate has a diacetyl content, which are at the range of between 1369 to 2668 milligram per gram. This is the range of what this diacetyl contain in a distillate. Okay, now the next is your advantage. Result in yield of sweet buttermilk, greater freedom when optimizing the temperature, treatment of cream before churning, yield more consistent product quality in a particular as a regard order and flavor. And there if something has an advantage, then they also have a disadvantage. Now, when a pure synthetically prepared diacetyl are added, then it give a undesirable taste of diacetyl. Harsh, even harsh, undesirable taste of diacetyl. Now, the next is your equipment which are used for a churning are called a churn. Now, we all know that butter is produced by a continuous process using a hygienically designed, cleanable in place. It is a continuous process. Okay, so churning process involve agitation of the cream at low temperature. Okay, so churning is what? Churning is the cream at low temperature give a result in the separation of the fat granule from the aqueous phase of the cream. Now, the buttermilk which are present in the aqueous phase is drained off, which contain most of the microorganism present. Okay, so the most of the microorganisms are present in where? In present in the buttermilk. It properly pasteurized cream while viable yeast and mold found at their present in butter is due to the contamination from the equipment, particularly the churn. Okay, so we all know that what should we do? We are using the pasteurized cream. Okay, now the butter are contaminated due to the equipment which is used for the churning. Now, churn are made up of stainless steel, so have their own regime of cleaning practices which ensure a clean churn. Okay, suppose that this is your churning machine. Now, we all know that if the debris of the cream um like leave out, okay, then the chances of the microorganisms will increases. Growth of the microorganisms will increases. Okay, so the next is your washing, salting, and working. In a batch type process, pasteurized chilled water is added to the butter grain in churn for washing purpose, which is used for removing a off flavor. Water, if untreated, may be source of contamination, mainly Pseudomonas species. Okay, if the water is untreated, then there is a chances of the contamination of the microorganisms, and mainly it is contaminated by a Pseudomonas species. The microbiological quality of water used for a washing or for a brine is critical to the production of safe and stable product. Okay, so water with less than 100 CFU per ml is a safe. Uniform distribution of this water droplet is carried out by a process called a working. Working of a butter little quantity change in microflora. There will be a 10 to 18 billion droplets of water are dispersed in 1 gram of butter, which are present in a water in oil emulsion. Now, water of butter originate principally from two sources, that is buttermilk, which include butter granules, and wash water remaining in butter. Okay, now the next is your inadequate working leads the formation of the moisture droplets which are less than 30 micrometer, resulting in a defect called a leaky butter and a color defect called a mottling. Another step in a butter making is salting if the table butter is to be prepared. Salt is added to give a concentration of butter around 2% in a butter, which is equivalent to the 11% in a serum. phase. Now dry salt is very difficult to handle, so either a saturated brine or for slightly salted butter around 50% suspension for a salted butter is used. Now the next is your packaging. When everything is done, now packaging is very important, because there is also if this packaging are not done in a proper hygienic condition, then there is a chances of the contamination of the microorganisms. Once the butter has been worked sufficiently, it is packed typically in a 250 gram of parchment paper or foil laminate, or into a 25 kilogram of polyethylene lined cardboard boxes for a storage. Okay, so either it is stored in a 250 gram of parchment paper or a foil laminate, or either it is stored in a 25 gram of polyethylene lined cardboard. During the packaging, contamination of microorganism is in butter mainly occur due to air, packaging material, unhygienic packaging equipment surfaces and personal, or slight growth will occur held at a temperature below 0 degrees Celsius, and none is expected at minus 15 degree Celsius. Okay, if the this butter are stored at 0 degrees Celsius, then there is a slight chances of the growth of the microorganism. But if the butter are stored nearly or less than minus 15 degrees Celsius, then there is no chance for the growth of the microorganism. Ideal temperature for the storage of the butter for keeping quality around 12 to 18 months, then the temperature should be at the range of minus 12 to minus 18 degrees Celsius. Okay, now the next is your microbial deterioration, spoilage of butter. Okay, if the growth of the microorganisms are occur, then how it affect the quality of the butter. Okay, the first thing is the color defect. Okay, its color will be defect, its flavor will be defect. Discoloration of butter maybe leads to the bacteria, yeast, and mold. Black discoloration like grease, smudge, it is caused by a microorganism, that is your Pseudomonas nigricans, due to a butter stored at a low temperature. Optimum for the pigmentation is 4 degree Celsius. Now the next is your fungal discoloration. Butter get discoloration due to the surface growth of mold, and defect is also described as a moldy butter. If the fungus grow will occur in a butter, then it will favor by a higher moisture content and acidity. Some common type of fungal discoloration frequently occur in a butter, which are as follow, like mold discoloration, like if the black, then it is caused by a cladosporium, e.g. uh, example your cladosporium, harbarum, aspergillus, haspodendrum, alternara mocher, rhizospores. And if the brown color are occur, then it will cause by a aspergillus species and phoma species, like muddy brown. And if green color and blue color are occur, then blue green color are occur, then penicillium species and aspergillus species are responsible for this defect. Orange and yellow color are occur due to the Gutoricum candidum, and reddish pink color are occur due to the Fusarium. And yeast will cause two disorder, that is your black and pink. Black are occur due to the Torola species, and pink are occur due to the Rhodotorola species. Okay, now the next is your flavor defect. Okay, so flavor defect means that either they are rancid, putrid, or cheesy odor is the most common flavor defect occur in a butter. The defect like malty, chunk like flavor, yeasty, and may also occur in a butter. Rancid flavor is of the butter are occurred due to the butter get rancid flavor due to the microbial, enzymatic, or chemical degradation of the fat. Okay, this fat will be degraded either microbially, enzymatically, or chemically. The hydrolysis of the fat in a butter is mainly due to the activity of the microbial lipases. Now this is the some example of the bacteria, mold, and yeast. Like okay. Now the next is your Putrefacive tent defect is due to the breakdown of the protein by various putrefactor organisms like Pseudomonas, putrefaciens, coliform, Flavobacterium, malorosis. The chemical compound produced during the breakdown of protein is closely related to a isovaleric acid, which is responsible for off flavor. Okay, cheese tend cheese like flavor in butter is occurred due to the association action of different gram negative rod shaped bacteria, which occur due to the storage of the butter at above 10 degrees Celsius. Other flavor tend like malty flavor is occurred due to the presence of this lactobacillus lactis were maltigenous in cream. The compound 3-methyl-butanol in butter is mainly responsible for the malty flavor. Shunk-like flavor is occurred due to the Pseudomonas membetica. Fishy-like tend occur due to the Pseudomonas ichthysomia and Deutericum cadmium and yeast due to the decomposition of the lecithin to a trimethylamine by a microbe. So this is all about the microbiology of the butter. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or for further query you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or for further query, you can also visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.